Thank you so much. I'm going to talk about a project that I did when I was still a PhD student in Juliana's lab, and I already want to mention the other people who were part of this. So this was co-led by Sebastian and me, also a former PhD student, and Morrissey, who was a master student in the lab, and obviously guided by Juliana. And we have this now as a preprint if you're interested in more details. And as a little introduction, as you probably all are aware of, that artificial neural networks are really powerful tools nowadays to learn to solve a specific task. For example, like this classical MNIST task where you have handwritten letters, and then the task is to correctly classify that this is a zero. However, if you have these artificial neural networks and you have a second task, for example, identifying handwritten letters, very often there's this problem that these ANNs actually forget how to solve the first task. It's called catastrophic forgetting. And so one question is really how to achieve continual learning without catastrophic forgetting in ANNs. And then on the other side, you have neural circuits. And as we all know, right, the brain is actually very good at you can learn one task and you don't have any problem in learning another task. And so the question is here, what are actually the mechanisms that allow neural circuits to have continual learning without catastrophic forgetting? And our project is somewhere in between. So we propose a biologically grounded model that explains how the brain can flexibly learn and combine new information to perform real-world computations while actually preserving this acquired knowledge and don't suffer from this catastrophic forgetting. And the biologically grounded features I will use, I will just introduce here briefly. We now actually heard a lot about assemblies in the previous talk. Just again, the definition is it's a group of strongly connected neurons and the idea is that they're really the basic building blocks of representations in the brain. And specifically, they can be learned by experimentally verified Hebbian plasticity. And there's a lot of literature on this. We actually recently reviewed this if you're interested. The second feature we're going to use, also what we heard in the previous talk, are dendrites. And in this case, we use dendrites as units. So dendritic branches as really independent, nonlinear computational units. So what I will mean by that is that you have different dendritic branches and they can receive inputs from different features. And the last biological plausible feature we will be using is inhibition as a gate, specifically saying inhibition can control the excitatory activity and the plasticity at the dendrite through a disinhibitory motif. So for example, VIP inhibitory neurons can inhibit SST neurons, which effectively disinhibits the dendrite. And there's really nice experimental work from the Oswald lab showing that this can gate plasticity on and off. And so in our project, I will now present kind of bridging of scales. I will start with the single cell model, and then we will put this single cell into a recurrent network, and then we will put many networks together and think about the computation. So let's start with the single network. So in this case, we now have 6 dendrites, and each of these dendrites can receive excitatory and inhibitory input. And important feature is that we model the dendrites having nonlinear NMDA receptors, and the soma is leaky integrate and fire. So we have spiking neurons. We take the model parameters from previous studies, and we also have plasticity, voltage-dependent plasticity at the dendrites. Again, take parameters from the previous studies. And what we can realize now is the following. So if you look at a single dendrite, and here I'm showing you in red, it will be long-term potentiation. So the excitatory synapse can increase in strength or depression in blue. And I show you this as a function of the inhibitory firing rate and the number of highly excitatory inputs. So basically as a function of inhibition and excitation, right? So imagine you're here at this point. And in this case, here you see the dendritic voltage, and the plasticity rule actually will slowly lead to an LTP with this amount of excitatory and inhibitory input. But just removing one excitatory synapse can actually move you now into an LTD regime. So you see slowly over time, the synaptic strength would actually decrease. And further reducing the input would actually not now give you into a no plasticity regime. And the same is true if you now increase inhibition. So you go from an LTP to an LTT to a no plasticity regime. So it's really about the realization that the relative strength of excitation versus inhibition can control the sign and the amplitude of dendritic plasticity. Okay, so now in our model, we actually have 6 different dendrites, and each of the dendrites can receive input from a distinct inhibitory distinct inhibitory neurons. And we call these the inhibitory, and we call this inhibitory contextual signals. And this is, as I mentioned before, inspired by this VIP SST disinhibitory pathway. So whenever I will say that, for example, a certain context is on, that means inhibition is on here, and these inhibitory cells are off, which means there's disinhibition or no inhibition at this specific dendrite. And now let's again see what happens with the synaptic plasticity at the excitatory cell. So for this dendrite number 1, which is now gated on by this specific context, there's actually no plasticity because it only receives weak excitation. So if we now switch on these groups of neurons, context number 2, switch off these groups of inhibitory neurons, we're actually now in an LTP regime, which you can see here. So the synaptic weights onto the specific dendrite will actually increase. And again, switching off another group of neurons, switching again the context, in this case, the excitatory strength is intermediate, so this will induce LTD. And you can go through this again. Again, there's no plasticity for the context number 1, there's LTP for context number 2, and LTD for context number 3. And importantly, if there's another dendrite that receives strong excitatory inputs, but it's never gated on, there's no plasticity here. So kind of to summarize this, the idea is that dendritic-specific gating of synaptic plasticity can be achieved via disinhibitory contextual signals. Okay, so now let's take these neurons and embed them into a network. So now we have 400 of these multi-unit neurons in our network, and we connect them all to all weakly. So somas are connected to dendrites. And we also add a feed-forward input drive. So we have a random subset of cells here in this feed-forward area that drive by sparse and random connectivity, drive a random subset of cells in the recurrent circuit. And again, we now choose one context to be on. Let's say it's the green context. And by chance, you will have certain, so this is just a spiking raster. So we have these feed-forward neurons, and a random subset of recurrent neurons will be spiking highly. And now just by chance, you will have a certain dendrites which are in the LTP regime because they receive a lot of excitatory input, and we will have certain dendrites that are gated on that are in an LTD regime or a no plasticity regime. And now the question is, what does happen to all the excitatory connections in the recurrent circuit? And crucially, only the within synaptic strength increases, which means only the neurons that have LTP-gated dendrites will actually have strong connections among them, and this is what we then call an assembly. So there's strong within connectivity. And together with feed-forward inhibition and recurrent inhibition to stabilize the system, we can actually show that you can stably learn these assemblies in the recurrent circuit. But importantly, the learning really only happens at this one gated dendrite. And again, to summarize this, we can show that there's stable assembly formation through dendritic inhibitory gating. We can also imprint multiple assemblies in the same context, but we have this in the paper. Okay, then let's move to multiple areas. And in this case, now we have these 3 networks, basically. We have area X, X prime, and area Y. And we want to now learn certain computations of assemblies. So the first thing we'll do is we'll learn a projection. So area X projects to area Y. And again, so we now have a certain random subset of cells in area X that are spiking highly. X prime now is just having spontaneous background activity. And a certain subset of cells in area Y will spike highly, and these will become an assembly. In addition to learning a projection to area Y, we can also learn an association. So now we activate actually a random subset of neurons in X and X prime. And again, this will lead to an assembly, strong connected groups of neurons in area Y, but this is now an association of these 2 assemblies. And now we can switch the context. So a different subset of dendrites will be gated on. And crucially, even if you activate the same set of neurons that we activated before, this means that in area Y, since a different group of neurons will be dendrites will be gated on, you actually learn a distinct assembly, as shown here. And the same is true for the association. So activating the exact same neurons, but being in a different context, because different dendrites will be gated on and different neurons will fire high firing rates, you have a distinct assembly here. And Another thing we then tested is if we now choose to have as many overlapping neurons in area Y than for the first assembly, by choosing them here in area X, but basically to induce a large overlap here. And again, you can learn an assembly even though it's largely overlapping with the green one. And the same is true for the association. Exactly. And so to summarize, this is really crucial. So we can have a complete neural overlap in the input areas X and X prime, and that leads to distinct assemblies in area Y if we switch the context. And it's also possible to have a large overlap of assemblies in area Y, again, if the context is switched. And crucially, we now don't have forgetting. So how you can see this is that basically the synaptic weights that have previously learned do not get erased by learning new assemblies. And this is one example to show that we can do some assembly computations. And so these ideas that you can really combine assemblies by having these projections or associations. And basically my last point I want to make is I want to kind of show an example how these computations might work in like a real life problem. And so in this case, we now have 5 areas and we have an auditory pathway and a visual stimulus pathway. So for example, there's a visual stimulus projecting to a visual sensory area. You could think about this as like thalamus, and then to a visual area, for example, primary sensory cortex. And then together with the input from the auditory area, it arrives at a concept area, some higher order brain region. And what we now do is we actually, as a visual stimulus, we use the MNIST dataset. So for example, this is a 1. We filter this by Gabor patches, and then we allow then these input is projected to a visual sensory area, into the visual area, and again, then forming an association with the auditory area, because at the same time, we actually present the auditory stimulus. In this case, this is 1. And so in this case, you would learn an association in the concept area by the mechanisms I explained you before. And then we have a second context. So we have now a number context and we have a letter context. And now we choose as a visual stimulus actually the letter L. And we chose this because there's no way, to be honest, to distinguish the visual stimulus of the number 1 and the letter L. But you know that based on the context, you are in a number or in a letter context. And specifically as well, the auditory stimulus is very different between 1 and L. So basically what happens is, in this case, you have a large overlap in the visual area, but in the concept area, as I've shown you before, these two assemblies will actually be distinct, even though the visual stimulus is very, very different because there's a different input from the auditory pathway. And we do the same thing for the letter O and for the number 0 because they're also highly overlapping. And then we present randomly different visual stimuli over a long time, and we learn these assemblies. And basically, this shows how in the concept area for each of these different stimuli, we form a distinct assembly. And as a last point, I also want to show that we can also do here pattern completion, which I think is interesting. So for example, now imagine we don't show any auditory stimulus, but we only show one visual image, for example, the 0 here. If you're in the visual area, there's no way to know if the stimulus is a 0 or an O. That is indicated here. So the firing rates of the assembly of 0 and O are basically the same. It even looks like the O is higher here. But in the concept area, because these assemblies are so distinct, it's actually very easy now to distinguish that this is a 0 or a O if you know the context that you're now in the number context. And so this is also true for the other visual stimuli, but basically showing here that we can do stable learning in this example of a visual auditory association task, and that we can have pattern completion when presenting only the visual or auditory stimulus in the concept area. And with this, I'm already at the end. I would just want to summarize what I told you. So I started out with this single neuron multi-compartment model. And we realized that excitation versus inhibition can determine the sign and the amplitude of plasticity at the dendrite. And we use this form of the dendrite-specific gating of synaptic plasticity by these inhibitory contextual signals. What I didn't show you is that the nonlinearity of the NMDA receptors is actually important, controlling the plasticity regime. If you're interested in this, check out the preprint. And then we move to the recurrent circuit, and we would show that we can learn robust assemblies. and that contexts actually allow large overlaps of assemblies, and there's no forgetting of previously learned assemblies. And again, in the paper, we also show that we can do robust recall and pattern completion of these learned assemblies. And finally, I showed you some example of possible assembly computations based on these projections and associations. And I had this specific example of the auditory visual association task. And then also in the paper, we have interesting further results of how recall is enhanced if you disrupt previous areas. And we also do associative learning via existing assemblies. If you're interested, please check the preprint. With this, I'd like to thank again my co-authors, my funding, and you for listening. Happy to take your questions.
Okay, thank you very much, Christoph. We have a couple of questions in the Q&A.
Okay.
So the top voted, I think I could combine the first two, is
Yeah.
on the experimental evidence for this. So what is the experimental evidence for dendritic branches? And the second question is, how could you test this experimentally, this model?
Right. So to your first, there's actually clear evidence in hippocampus at least that different dendritic branches can be used independently by the Basel lab. I had the reference at the beginning, where they can actually really show that at least different features arrive on different dendrites. Then there's also a lot of evidence coming more and more that there's like synaptic clustering. So not thinking about like a dendritic branch by itself, but that features, synapses that code for the same features actually clustering on the dendrite. So I think that's also one way to think about this. And then the second question was
how could you test this experimentally? This is probably a hard question, I guess.
Right.
As
I think
always.
it would really, because it would really depend on the context, right? So what we kind of suggest is that depending on the context, a single neuron can code for multiple features. I think we know that neurons are tuned to multiple features, right? We know that. I think it would be interesting to really show the direct link that different dendrites receive different inputs, and that this is actually the reason why there's tuning to multiple features of a single neuron. I think that's one thing I would suggest that at the single neuron level.
Mm-hmm.
Yeah.
Okay. And then another question was on the how strict this model is with respect to how this context is represented. Because if I understood correctly, also you provide now really like a binary binary context. It's on or off, right? Could you have graded contexts in there?
Yeah. I think that's an excellent point. I think that's how I would kind of moving forward, trying to improve it. We basically choose the context, right? So we say we have 6 dendrites and we have 6 different contexts. I think there are multiple ways to improve this, to think about random contexts and, as you said, maybe like graded context, or think about how one could actually maybe learn these inhibitory synapses, right? Thinking about Tomoki's talk, for example, with inhibitory plasticity, is there a way to be able to learn these specific contextual gating mechanisms? I think that would all be really great. But yeah, in our project, we just basically put it in by hand. And, yeah.
Okay, so far you haven't tried this, so I think that covers this question. How do you maintain EI balance in the network? Do you have any homeostasis in there?
Yes, exactly. So that's actually very important, especially because sometimes we learn projections, sometimes we learn associations. And if you do an association, you suddenly have double the excitatory input into one area, right? So what we have is basically we have feed-forward inhibition and we have recurrent inhibition mechanisms. So this really keeps the firing rate of each of these networks stable. And it's like a key to avoid assemblies becoming too large and getting into these unstable regimes. Yeah.
Okay, I think maybe I can very quickly ask one question for myself.
Sure.
So you had these hierarchies in the model, right? Do you know what would be the depth of such hierarchies that you would expect in a biological brain? Is there any evidence for this? How many levels? Because I think you have 2 levels now, but in principle, this could be any number, right?
In principle, it could be any number, exactly. And also our approach could be used for basically any number of like hierarchies. So then it depends. Yeah, I think it also depends on what animal you look like, right? You have primary sensory visual cortex, V2 or V4, depending on the animal, right? And then higher order brain regions where you maybe want to, like we have in the concept area where you try to associate different concepts. I think for our modeling framework, there's no limitation of how deep the hierarchy is. How it is in the brain, I think it really depends where you're looking at, to be honest.